Forms of damage, radiation damage to cells that can include damage to the DNA of the nucleus, damage to the cell membrane, so it can leak fluids or lose nutrients, damage to the mitochondria, which interrupts the food supply, or damage to these other things which result in the cell digesting itself. But the main thing that we're interested in is DNA damage. So that's the main issue that we deal with in radiation protection. So damage to the DNA, this can be damage to the bases. It can be crosslinks between various strands of DNA, protein crosslinks, intercalation, but the main thing is these single strand breaks where one of the sides of the ladder gets broken, or double strand breaks, which are even more important, is where both sides of the ladder are broken at roughly the same spot. These can be reversible and repaired, and the DNA does repair itself, but the greater the damage, the less likely that the repair will be error-free. So the outcome might be the death of the cell, or malignant disease, or even genetic damage. So double strand breaks need a break in both strands of the DNA about 2 nanometers apart. Lesions can travel, so breaks can travel along pairs, but it still has to be within a few nanometers. And the likelihood of this occurring depends on the type of radiation and the radiation dose. The number of single strand breaks and base damages per unit of radiation is about 2 orders of magnitude larger than double strand breaks, but it's actually even though double strand breaks are occur a lot less frequency, they're the ones that are most important. So the double strand breaks correlate best with cell killing. So let's have a bit of a look at what happens initially here. So we can have what's called direct effects or indirect effects of ionizing radiation. The direct effect is where the ionization event occurs in the actual molecule of importance, so it actually occurs within the DNA itself. Whereas the indirect effect, the damage occurs in water around, sitting around the DNA, so actually in water, which then produces free radicals, which then damage the DNA. So direct effect directly damages the DNA, indirect effect damages water, and the water goes then to damage the DNA. So this occurs very quickly, 10 minus 15 seconds, and free radical formation occurs within 10 to the minus 10 seconds. Here's just a picture of the different types. So the direct effect is incident radiation directly affecting the DNA, where indirect incident radiation affects a water molecule, which then forms a free radical, which then damages the DNA. Either way, the DNA is damaged. So the indirect effect of ionizing radiation occurs through this for people with a chemistry background. So more than 80% of the human body comprised of water. So radiolysis of water results in the production of free radicals. So we've got a water molecule plus a gamma goes to form these, which goes to form these and follows this chemical reaction here. The free radicals, they're the uncharged molecules containing a single unpaired electron. So these are highly reactive, and these will attach to the DNA. So these are basically the free radicals that will cause damage. The longer the lifetime of the radical, the further it can travel. Hydrogen peroxide is a free radical, so that's an important product in radiobiology. Many of the processes of radical formation are enhanced in the presence of oxygen. So there's an oxygen enhancement effect. So if you have oxygen in a cell, it will be more sensitive to radiation than a non-oxygenated cell. And we'll cover this again, we'll cover this later on. But this leads to the oxygen enhancement ratio, or the OER, which is the cell kill, there's a word missing there, cell kill in the presence of oxygen divided by cell kill in a hypoxic environment. And typically it's about the order of 3. If you have oxygenated cells, they're 3 times more likely to be damaged. So the free radicals are very reactive and damaging. About a third of the damage is caused by X-rays, low LET ionizing radiation, so we'll cover that in the next part of this lecture. About a third of it is due to the direct effect, but the indirect effect causes about 2/3 of the damage, so the indirect effect is more damaging. The human DNA molecule, that's about 2 meters long, but very narrow, and to pack this into a nucleus with a diameter of less than 10 microns, the DNA sort of gets wound up in all these structures here. So this is a progression of the winding up of DNA, and it eventually looks like that. But what this basically means is that the DNA in here is very, very tightly packed. So if you get some radiation interactions close together in there, you're highly likely, or much more likely, to cause a double-strand DNA break. So DNA is some superstructures that are illustrated there. And so a long-lived or far-ranging radical can have many opportunities to interact and be much more likely to cause damage. DNA is the largest molecule in the cell, and it takes up about 10% of the cell space. So the DNA configuration changes through the cell cycle, so that's why the cells are more sensitive at some times than others. But even when not cycling, the DNA can be damaged.